Today we're meeting with Dr. Boynton at the University of Washington. He studies a variety of topics concerning visual perception, attention, and fMRI methods. Thank you for taking the time to meet with us.
Well, thanks for having me.
Could you talk a little bit about the relationship between attention and visual perception?
Well, that's a really tough question. Of course, that's what I do for a living, so it keeps me busy. Back when I was in grad school, I was taught that attention and perception were really separate processes, where perception is really the process of breaking down a visual scene into its components, say objects and faces and things like that. Attention is the process of pulling out the important things from that breakdown to things that are relevant to some sort of action. But more and more recently, we're seeing that these two processes are very closely related.
And how are they interrelated?
Well, I think the big breakthroughs have been through neurophysiology experiments maybe 10, 20 years ago, where people were measuring measuring responses of neurons in early visual areas such as motion processing area, MT.
Could you maybe give an example of one of those experiments?
Sure. Actually, the experiment that kind of changed my life is a study by John Monsell, who was in Texas at the time, who came to visit Stanford, where I was a postdoc, and he gave a talk on measuring responses of neurons in macaque monkey area MT, which is in the back of the head. Area MT neurons are very selective to motion. A given neuron will say like upward motion very, very strongly. At that point, there's a lot of research studying this part of the brain and monkeys presenting stimuli, different directions of motion, and measuring the response properties. It was really believed that these neurons were simply acting in a feed-forward sort of image processing fashion.
What do you mean by feed-forward image processing?
So, in fact, a lot of these experiments up to that point were done in anesthetized monkeys, monkeys that were asleep, and they didn't think it really mattered because the visual system was just responding to the input. And didn't matter what the monkey was awake or not, or what the monkey was paying attention to. But these studies by Monsell and others, and Desimone and Duncan around this time, were surprising because they showed that what the monkey was asked to do about this visual stimulus, pay attention to one particular direction of motion, say, strongly affected the way these neurons fired. To the point where you can make a neuron fire 10, 20, 30% stronger if the monkey was paying attention to, say, upward motion than downward motion.
Just paying attention to that?
That's right. So the classic experiment was they presented two dots in the receptive field of this MT neuron. This is the part of the visual field that this neuron likes. Two dots that moved in opposite directions. One was always moving up and down and back and forth like this. Suppose this neuron prefers upward motion. So when these two dots are doing this, there's always one dot moving in the preferred direction of the neuron. And so you'd expect just a constant response of this neuron in spikes. Bzz, 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 bzz. But what they did was had the monkey pay attention to one of the two dots. So say this dot right here in my left hand. And when they did this, only when the dot that the monkey was paying attention to moved in the direction the cell liked, the neuron would fire. So it would go bzz, bzz, bzz. So even though there's always one dot moving in the preferred direction of the cell, only when the attended dot moved in that direction did the cell really fire. And this is a big deal because we thought that these neurons didn't care at all about what the monkey was thinking about. So really 3 synapses away from the eye, we're seeing a huge modulation in the response of these neurons to what the monkey is paying attention to. And this kind of revolutionized the way we think about attention versus perception. Because if we're modulating the responses of neurons this early in the processing pathway, we're really changing effectively the representation of the stimulus in the brain, really effectively the percept of the stimulus.
Does that work for other visual features such as contrast or orientation?
Well, it does. And so since then, there's been a whole bunch of experiments done in both monkeys and fMRI in humans, for example, trying to figure out how early in the visual processing pathway can attention modulate these responses.
And how early is that?
Well, so work that the last experiment I did as a postdoc was to study, was actually kind of interesting. We were really inspired by this monkey experiment with motion in area MT. We said, okay, let's try to see if we can find this in humans. And so we spent a whole summer training ourselves on these motion discrimination tasks in the laboratory. Because this was crazy. Area MT being affected by attention, this can't be. And so we think, well, one reason why this might be happening is because these monkeys that John Monsell were using were professional monkeys. I mean, their job was to pay attention to one dot and ignore the other one, and they spent their lives doing this in these experiments. And so we thought, well, maybe their brains are slightly different. The reason why MT is modulated in these monkeys is because, they're not normal. They're overtrained on this task. So this undergrad and I, Sunil Gandhi and I, spent the summer in the dark doing visual perception experiments for motion discrimination in a particular part of the visual field for the whole summer before we did our first fMRI experiment, because we were trying so hard to make sure we got attentional effects in the human homologue of MT.
And did you find the effects?
We did. So the experiment was very simple. It turns out that spatial attention experiments are ideally set up for fMRI. And this is because when a visual stimulus is presented in the left side of the visual field, the responses happen in the right visual cortex, and vice versa. Exactly. And fMRI doesn't have great spatial resolution, but it's good enough to pick up separate responses to different sides of the visual field. So you can do a really simple experiment. Have a subject lie in the scanner, fixate straight ahead, and present, say, two fields of moving dots, one to the left and one to the right side of the visual field. And have them pay attention to the left thing, make a decision about, say, how fast those dots are going, and ignoring the right one. Do that for 20 seconds. And then switch attention to the other side and go back and forth. Importantly, you keep the visual stimulus constant through the whole experiment. The reason for that is, with fMRI, we're measuring changes or differences in responses rather than absolute levels. We have to always compare things.
So you need a baseline response for one side versus the other.
That's exactly right. And so if attention didn't affect anything, then if the subject was basically staring at a constant stimulus, visual stimulus, then we wouldn't expect any modulation in the responses in the brain. But what we found was, when the subject flipped their attention back and forth, left and right every 20 seconds, there was a modulation, the fMRI response, in the contralateral side of the brain. So there's a bigger response to the attended stimulus.
So how do they, you can actually adjust your attention without moving your eyes too. You can stay focused on one specific point. Yeah,
that's called covert spatial attention. And it's a little unnatural, but with practice you can get good at it. It's very natural to go look at the stimulus you're paying attention to. But with enough practice, you can fixate really carefully. And we had eye trackers to make sure that people weren't cheating.
So you just throw away any result whenever somebody's eye actually moves. You just get rid of it.
They do the same thing with monkeys. They track their eyes very carefully, and they throw out the trials where you don't look, where you actually cheat and look at the stimulus.
Okay.
And so this experiment, we went in. We finally ran the experiment at the end of the summer, and we looked at the data, and sure enough, we found big modulations in area MT, the human homologue of area MT, HMT+, for the attended stimulus. But what was most amazing is we also found about the same strength of modulation in V1, the primary visual cortex. We did not expect that at all.
And so that's really early in the visual stream.
That's right. So that's 2 synapses away from the eyeball. And that is, we think of the V1, or the primary visual cortex, as the sort of the gateway, or almost the bottleneck for visual processing. Conscious visual processing really kind of passes through V1 and then spreads out to higher visual areas. And so if modulation is happening with attention in V1, that means that really the representation of the stimulus is being affected everywhere in the brain.
So would that be true for all people, regardless of, like you said, you spent an entire summer training for this one experiment. If you just took a naive participant, would you find the same results?
Exactly. So we were naive about that. It turns out that it doesn't matter how much training you have. I could take you, train you up for 5 minutes in the laboratory to how to not move your eyes when you're paying attention to something, throw you in the scanner, and your results would be just like mine after the summer. So these learning effects were not a big deal. These effects, these attentional effects in V1 are robust, and they're happening all the time. Interestingly, since then, there's been experiments by Sabina Kastner at Princeton showing attentional effects with fMRI in the LGN, which is 1 synapse away from the eyeball. And so that's how far we've been able to push this, is within 1 synapse away from the eye, attention really seems to be modulating the representation.
But there is no attentional effects in the retina whatsoever.
Well, we joke about that, right? And we used to joke it was, haha, it'll never be in the LGN, but sure enough, it's there. There are some slow, weak feedback connections to the retina, but they really seem to have to do with light modulation and circadian rhythms, so we don't think that there's enough feedback to really modulate the effect, the response in the retina to attention. So I think we're safe there.
Okay. So what sort of mechanisms underlie or behind attention?
The basic results of spatial attention are straightforward. When attention is directed to a particular location of the visual field, neurons that have receptive fields in that location seem to have a higher firing rate when you present a neuron. But if you do more interesting things, like present multiple stimuli inside the receptive field of a cell, say like those two dots within the same receptive field, more interesting things start happening. It matters which component of that stimulus you're attending to. So, for example, if you have a neuron that, say, prefers upward motion, and you present two patterns of motion in the receptive field, one up and one down, if you have the monkey pay attention to the downward motion, the direction that the cell does not like, you can actually have a suppressed firing rate of this neuron versus if the monkey's paying attention elsewhere. So it's not so simple that the firing rate just goes up. It really has to do with what feature the animal, monkey or human, is paying attention to.
In some of your articles, you talk about contrast gain. What is that precisely?
So I mentioned that if you attend to a particular location in space and the neuron has an increased firing rate, the important question is, well, what would you mean by an increase in firing rate? How does that interact with, say, the strength of the visual stimulus? So, for example, if I put a low contrast stimulus up, which barely makes the neuron fire, and I attend to that, versus a very strong stimulus, a high contrast black and white flickering stimulus, how do these effects of attention depend on the strength of the stimulus itself?
You mean the level of the effects of attention? So the size of the baseline increase or the firing rate increase?
Kind of. So we know that most visual neurons respond stronger for higher and higher contrast. They have sort of a contrast response function, as we call it, that goes up and kind of saturates and flattens out. So an important question is, well, if we add attention to the stimulus, how does that affect this curve? Contrast gain is the idea that when you pay attention to a stimulus, it effectively increases the contrast of the stimulus. In other words, if I have a 20% contrast stimulus, contrast goes between 0 and 100%. If I have a 20% contrast stimulus and I don't pay attention to it, I get a certain response. If I attend to it, I'll expect to get a bigger response. Maybe it'll act as though it was a 30% contrast stimulus.
So by attending to something like contrast, you're literally changing the way you perceive that visual feature?
Well, that's an interesting question. So contrast gain is the idea, like I said, is you're effectively increasing the contrast of the stimulus by attending to it. It's appealing that way because you're sort of just turning up the knob of the strength of the stimulus. And it directly addresses this question you have about attention versus perception. It's appealing, but the problem with it is that it doesn't really follow the way we perceive the world. When we attend to something, it doesn't look like it has higher contrast. Right? Attention is really defined by our behavioral measurements. If we attend to something, we know we can respond to it faster, we can discriminate things better. But if you apply spatial attention around the visual field, you don't feel like it's getting more high contrast when you move your attention around.
So it just makes you more aware of the contrast, not necessarily like it doesn't make it more pronounced?
That's a really good question. We just don't know. And like I said, if attention can boost the responses of neurons all the way through the visual system, then it would follow that it should make things look different, right? But it doesn't. And that's one of the biggest open questions in attention research, is how can we change the firing rate of neurons for an attended stimulus without changing the way it physically looks to us? And it just doesn't. We don't know.
That's very interesting.
I have no idea.
Another thing you talk about in one of your, in some of your articles is biased competition. What is that?
So like I mentioned before, when there's two stimuli within the receptive field of a neuron, one theory for how these attentional effects happen is an idea called biased competition. Desimone and Duncan and others have proposed the idea that if I have two stimuli, say one stimulus that the neuron likes, say upward motion, and one stimulus that the neuron does not like, downward motion, when presented together, these two stimuli effectively compete with each other to dominate the response of the neuron. And that competition is biased by what we're paying attention to. So if I pay attention to upward motion, biased competition would predict that this neuron will really respond as though only the upward stimulus was presented there, and it'll suppress the presence of the downward motion, and vice versa. If I'm paying attention to downward motion, the stimulus that this neuron does not like, neuron will act as though only downward motion was present, and it'll have a very weak response.
How do the older models developed by Anne Treisman and Broadbent relate to our more current views on attention?
Well, they've sort of been incorporated into what we know about attention now. So there was this big debate about early versus late selection. And it became sort of ambiguous what we meant by that in terms of early and late. Do we mean early in time or early in the visual processing stream? And once we realized that there's all this feedback and loops and things in the visual system, then these ideas really became kind of muddled and confused. But it's sort of come back in the sense that now that we've found attentional effects really early in the visual processing stream.
You mean as early as LGN?
Exactly.
Yeah.
That sort of brings back the idea of early selection, that we're selecting on information very early in the stream. But it is confounded with the idea of time. So one thing with fMRI, suppose you have an attentional effect measured in the LGN. Well, we know that the LGN is responding more for an attended stimulus than an ignored stimulus. What we don't know is when these effects are happening. One idea is that maybe the initial response to a stimulus is not modulated by attention, just blasts on through, like I mentioned, in a feed-forward fashion, and then it's attenuated in a later response through, for feedback loops affected by attention. This is something we can't measure with fMRI because the responses are so slow. And so the idea of early versus late versus is a little bit confounded when you talk about location in the brain and time.
So you talked about a lot about attentional paradigms. What happens when something pops out and grabs our attention?
Yeah, so far all the experiments I've talked about involve people or animals sitting there, told what to pay attention to, either location or a feature. But a whole other type of experiment or condition for attention is what's called an exogenous cue for attention, where something flashes or grabs your attention automatically. So endogenous cueing for attention is what I just mentioned, where you're told ahead of time what to do. Exogenous cue for attention is something, like I said, outside grabbing your attention. That's a whole other question about how that happens. What sort of stimuli will attract attention versus ones that you can completely ignore?
Are there different neural correlates for exogenous and endogenous cues?
It's kind of an open question. In fact, if you read the literature, it's a bit muddled. There are people that use exogenous cues for attention to study effects of attention in the brain, like of humans with fMRI, and they cite all the literature like that we use for endogenous cues, almost as though they're assuming these two mechanisms are the same, but they're probably very different.
What do you personally think?
So in experiments with exogenous cues, the main question is, what sort of things attract your attention? And of course, there's a long literature in the auditory domain about what sorts of things attract your attention. We know that if you hear your name, for example, it automatically grab your attention, these sorts of things. Visual system, a similar parallel research has gone on trying to figure out what grabs your attention visually. And some things are obvious. So the visual is called salience. If you basically take a visual image and break it down in terms of its color and contrast and things like that, regions of a scene that have highest contrast versus the background, more different color or different brightness or something like that, will automatically grab your attention. And we can use simple computer algorithms, just break down the image in terms of color and contrast and orientation to basically predict where a subject is going to look first on the scene. It's called a salience map. And it gets us a long way to what sorts of things grab your attention. And typically features that are encoded by neurons early in the visual system, such as orientation and direction of motion and say color, those are the sorts of things that are good for being highly salient for grabbing your attention. And so there's a lot of mathematical models out there about how these different properties like orientation and color compete or work together to create a salience map. And so people try to add different weights to different properties like orientation or whatever to try to predict where a subject's going to look when they're looking for something in a scene. But there's much more to it than that. We know that certain things that have same kind of visual salience as far as an image processing system is concerned will grab attention. A scene a graphic scene of violence or something like that, or a gun, we know those things will grab your attention more than a similarly shaped object that has no,
Semantic meaning.
Exactly.
How do exogenous cues engage attention?
Well, we don't know. I mean, what we've talked about so far are the effects of attention on processes in the early visual system. What we haven't talked about is what is controlling these things. Somehow there has to be some signal feeding down, telling this neuron to shut up and this neuron to fire more. What is going on there? That's a big open question in attention research, and it's a tough one. It's relatively easy for me to put somebody in the scanner and say, attend to this, ignore that, measure the response, publish a paper saying, yeah, V1 responds more when you're attending something versus not. It says nothing at all about what is controlling these things. What part of my conscious effort is how does conscious effort change the neural firing rate in V1? And same thing for exogenous cues. Why would a looming stimulus that's coming at your head, how does that automatically modulate, say, the responses of neurons that are representing the stimulus? We just don't know. I mean, one idea for exogenous cues is that there's some sort of map, and say a priority map, they call it, in say lateral internal LIP in the brain or maybe the prefrontal cortex, that is basically a map with neurons that represent different spatial locations, and basically there's a higher firing rate for a neuron that represents a location that we want to attend to. That's basically saying, and that's the result of all these different things we talked about before, salience versus behavioral relevance, having to do with whether it's going to hit your head or not. All these things are sort of incorporated into a central map that says this is where I'm attending. And that is somehow now feeding back in and modulating neural firing rates in the visual system.
So does this map act as a sort of homunculus that controls awareness? Sort of.
Of course, the homunculus problem is really a problem in neuroscience and in psychology, right? So if you've got a homunculus that's trying to control which neurons are firing more and more or less, what's controlling the homunculus? So it really isn't an answer to the question. That's one of the reasons why this salience map or this priority map isn't really an answer to the question, because something has to control this map.
Well, that was all very interesting. Thank you for taking the time again to sit down and talk with us today.
Oh, thanks for having me.